Dear technicians, today let us look at one of the hematology instrument that is present in Indiana Hospital. So that is Horiba Yumizen H550. That is our uh, routinely used hematology instrument. We also have a backup of Horiba Yumizen H500, about which we will make the video at the end. One important thing which we want to consider here is many of the students and technicians who come from other labs and all, when we are taking the interview, one of the thing they keep asking, when we ask, they don't know what is the name of the hematology instrument or the biochemistry instrument and all. It is very important that you should know. So this is Horiba Yumizen H550. So Horiba has many series. This is Yumizen series. H550 is the instrument. Okay. So this has got a batch analyzer also, as well as it has got a stat analyzer also. So this is a instrument where you can use you can give the EDTA samples directly without opening the cap. Okay, so this is the H550 instrument, which is not yet switched on. Okay, so this is the left side end of the instrument where all the reagents are kept, and this is the right side end. This is the area where you can give 10 samples at a time in a batch. There is an adapter which is present where you have to keep all the vacuum and then give. So now normally we don't use batch analyzer in this instrument, so we are not going to teach you about this immediately. This is the back end of the instrument. So this is the power plug. So this you should know always, because every night when we are switching on and switching off, this is the button which you need to switch on and switch off. This is the pipe for the waste channel. Okay, so these are all the reagents that are normally used in this instrument. This is the ABX diluent, which is about 20 liter capacity. This is the ABX white diff, which is 1 liter capacity. This is the cleaner, and this one is used only during some special wash, concentrated cleaning, what we call in one of the maintenance time. Only during that time this reagent is used. So normally to run the instrument we need only these three reagents. In this also, ABX cleaner is required only when we are switching on the instrument and switching off the instrument. So for running of the instrument you need only these two reagents, white diff and the ABX diluent. So make sure that those reagents are properly kept in that instrument. So this is the ABX white diff and this is the ABX cleaner. The reason why it is kept in this fashion is we can see from outside only what is the reagent level that is present. That is why it is kept in this fashion. This is one of the rules in the lab, to keep it in such a way that we can always see from outside what is the level of the reagent. Now one important thing which you should always know is the pipe. See, very important thing is this instrument works not with motorized pump, but instead it works on a syringe pump. So whenever syringe pump is being used, the suction of the instrument is not that good, but the precision of the instrument is very good. So because of that reason, it is very important that these reagents are kept at the level of the instrument. So if somebody remembers Sysmax and other instruments, you can see that these diluents are all kept down. It need not be at the same level of the instrument, but here in this case it has to be on the same side. Okay, that is one important thing. Second important thing is the pipe. Okay, now this pipe doesn't have a filter attached to it, and this part of the vertical part of the pipe is very hard and rigid. So it's very important that they are all the way down to the reagent. So this is also many times they do mistakes where the pipe is hanging on the top, which doesn't touch the reagent level at all. So this is also very important that whenever you start working with the instrument, make sure that these pipes are all the way down into the instrument. It should not be on the top like this where it is not touching the water level and all. Okay, this is very important. Okay, make sure that these pipes are all the way down, touching the lower end of the reagent. So once you switch on, immediately like this, the hard disk starts switching on. So also remember that this instrument works on Linux platform and it doesn't work on Microsoft platform. Many of the technicians may not understand why this is important, but over a period of time, when you spend some 4 or 5 years, you may understand what exactly mean by this. Okay, so this works on Linux computer. So once it starts switching on like this, various display changes will happen, and ultimately it will come to this level. So here as you can see, initially the username password, they're all not enabled, all are in the mask level. So it will go for checking of the synchronization. It will check for the whether the software versions are proper or not, and database of the instrument is proper or not, and then the initialization of all the moving parts of the instrument happens. Then subsequently it will allow you to enter the username password. Now username and password, the username is lab lab, password is also capital lab lab. So you can pick it up from here. So you click on this, immediately like this window will open through which you can enter, or you can even enter it through the keyboard that is attached. The reason why I specifically mentioned that it is working on Linux is because Linux systems doesn't have a mouse attached. So even this keyboard also we have attached as an extra. It is not a part of the instrument. So you have to work with this keyboard only, which is a on display keyboard, but we have attached the extra keyboard to our instrument. So from here also you can enter the username password. So once the instrument's global initialization is happened, so it will option the to validate so the username and password. So immediately once it is done, you can click on the validation button. So it will ask for another whether to reset the auto numbering or not. It is always advised that you click on this reset button, but never click on the erase or explore patient result option. Only click on the reset button. The importance of this also, over a period of time, as you start working, you will understand, and then you validate. So you click on this. So immediately this is the main menu of the instrument. So here it will be asking for startup. So like this, the startup will be blinking, and all other parameters, they are all will be in the mask. So you can't run the instrument now unless you give the startup. Now few things you need to remember, you can learn is the option of startup, where it will clean the instrument, run the diluent as a sample and see what is the results and all. This is the reagent window, quality control. So everything about quality control is there. Reagent, where everything about the reagent is there. Maintenance of the instrument, regarding any particular maintenance is required, that is there. Logs will tell you about the reagent logs, the any alarm logs or anything like that. Settings is usually not enabled to you, so only few settings only will come in this. So let us not bother immediately. Okay, so then you have to look for these four indicators. This is about quality control. So if any errors or anything is present in the quality control, this will be in red color. This is about the communication, means interface that has happened into the instrument, so that it will mention. So if any error is there, so that will tell, this will also be red. This is about reagent. Any reagent is getting expired, or it is in low, the volume is low or anything like that, so this also becomes red in color. This is system. System means any moving parts or any software or anything has got some problems, so this will get into red. So when everything is green, so you can go for a startup. So you can click on startup, but always remember that switching on and switching off usually is done in the night hours. So reagent additions also are done in the night hours. So if you have to change any reagents at this time, so don't give the startup immediately. After changing the reagent, you have to do again one more startup. So because of that reason, startup is done later. If all the reagents, everything is proper, you can go for a startup. So you click on the startup button, which will be blinking. So it will take another 5-6 minutes for the instrument to work. After that, it will be ready. So this will not blink anymore. Now you can see that it is a ready mode. So before it was mentioning here that there is a startup that has been not done. So all these things were not enabled. So once the startup and everything is over, so all these buttons will get enabled automatically. Now you should look for the what is the report of that particular startup. If any error is there, this would have become red. So if there is no error, means everything has gone well. Always click on this logs button. So there you can it opens like this. You can go for this blank cycle log. So you click on the blank cycle log, it will tell, see, this is the thing. So everything is green. So if the platelets were above 5, it would have blinked as red, and any changes in this also it would have mentioned. So it is clearly mentioning that it has passed the startup. So now you can start working with this instrument. Okay, so hope you are able to understand this. Thank you.